Hello, today I will be discussing the experiments of Nirenberg and Mattaei, and Nirenberg and Lieder. In the Nirenberg and Mattaei experiment, they wondered what amino acids are specified by codons composed of only one type of base. They began their experiment with RNA homopolymers poly-U, poly-C, and poly-A. They attempted to use G nucleotides as well, however they did not yield any results. These homopolymers were joined together by polynucleotide phosphorylase and combined with a cell-free translation system, which was extracted from a cell, a single labeled amino acid and 19 unlabeled amino acids. The tube was then placed in an incubation at 37 degrees Celsius. This was replicated 20 times with a different labeled amino acid each time. Under the incubation conditions, the tube contents began to undergo translation. Once translation occurred, the protein that formed was filtered out and checked for radioactivity. Once all 20 trials were performed, Nirenberg and Mattaei were able to determine that UUU encoded for phenylalanine, AAA for lysine, and CCC for proline. In the Nirenberg and Lieder experiment, they pondered if tRNAs were used, what other matches between codon and amino acids could be determined. To test this question, the pair combined the synthetic mRNA with one codon, 20 tRNAs with amino acids, and a ribosome. Once mixed, the ribosome bound to the mRNA and the tRNA to a specific mRNA. The mixture was then filtered through a nitrocellulose filter where the tRNAs paired with the ribosome-bound mRNA remained in the filter and the unbound tRNAs passed through. The filter was evaluated to determine the amino acid was bound. From this experiment, Nirenberg and Lieder were able to determine that when mRNA with GUU was added, the tRNAs on the filter were bound to valine. This experiment was also used to determine many other codons, which resulted in the discovery of 61 codons in total. Thank you.